Neurons. Under this topic, we will discuss the following: the cell body, dendrites, mitochondria, axon, and neuronal zones. The typical neuron consists of three main cellular compartments: a cell body, a variable number of processes that extend from the soma called dendrites, and an axon. The cell body. It is the main genetic and metabolic center of the neuron. It consists of the nucleus and nucleoli. Dendrites. Dendrites are tapering and branching extensions of the soma and are the main direct recipients of signals from other neurons. Dendrites have ligand-gated sodium ion channels that allow neurons to respond to chemicals released by other neurons, and their characteristic branching pattern allows for integration of multiple input signals. The dendrites have small knobby projections called dendritic spines. Types of dendrites. Primary dendrites are those that extend directly from the soma. Higher order dendrites are daughter branches extending from a more proximal branch, in which proximal refers to closeness to the soma. Individual dendrites can be longer than 1 millimeter or only 10 to 20 micrometers in length. Mitochondria. Mitochondria provide both a source of energy for processes associated with synaptic transmission and substrates for the synthesis of certain neurotransmitter chemicals such as amino acid glutamate. In addition, mitochondria contain enzymes for degrading neurotransmitter molecules such as MAO, which degrades catecholamines, and 5-hydroxytryptophan and GABA transaminase, which degrades GABA. Axon. The first part of the axon is known as the initial segment and arises from the soma, in a specialized region called the axon hillock. The axon differs from the soma and proximal dendrites in that it lacks rough endoplasmic reticulum, free ribosomes, and a Golgi apparatus. The smooth endoplasmic reticulum is involved in the intracellular storage of calcium. Smooth endoplasmic reticulum in neurons binds calcium and maintains the intracellular cytoplasmic concentration at a low level, about 10 to the power of -7. The axon divides into presynaptic terminals, each ending in a number of synaptic knobs that are also called terminal buttons or butons. In myelinated neurons, a sheath of myelin, which is a protein-lipid complex, is wrapped around the axon. In the peripheral nervous system, myelin is produced by Schwann cell, which wraps axonal membrane up to 100 times. The myelin sheath envelopes the axon except at its ending and at the nodes of Ranvier, periodic 1 micrometer constrictions that are about 1 millimeter apart. In the CNS, most neurons are myelinated. Myelin is produced by oligodendrocytes rather than Schwann cells. Neuronal zones. From a functional point of view, neurons generally have 4 important zones: a receptor, or a dendritic zone where multiple local potential changes generated by synaptic connections are integrated, a site where propagated action potentials are generated, an axonal process that transmits propagated impulses to the nerve endings, and the nerve endings where action potentials cause the release of synaptic transmitters.